Hey guys, we're going to go over the lesson for today about the properties of macromolecules. So we're going to think about which would we use, a fork, a knife, or a spoon to eat this soup. It's kind of obvious answer, right? We'd use the spoon, but why would we use the spoon and not the fork or the knife? And it's all about the structure is relating to the function of that utensil, right? So the structure of the spoon makes it have that function of being able to use for liquids. But if we were to change that structure, maybe to have it be have holes inside of it, it would have a different function. So the same thing is going to go for macromolecules, and that the structure is going to relate to the function of our macromolecules. So we're going to go into more detail of all of them. So our carbohydrates, as a review, the monomer is monosaccharides, polymer, polysaccharides, bond name, glycosidic bond. Its function, I'm going to scoot this over here, it is to have short-term energy storage. It can also be used for some structure. And its structure is going to be made of carbon, hydrogen, and oxygen in a 1 to 2 to 1 ratio. So let's go into those a little more details. So here are some pictures of some examples of monosaccharides. So mono meaning those are the monomers. Okay, there's different ones we could use, but these are all examples of monomers. So some of them are longer and shorter. They usually have 3 to 7 carbons. They are going to have a carbonyl group, which is this carbon double bonded to an oxygen that all of them have. And they're going to have names that end usually with ose, aldose, ketose, triose, pentose, hexose here. So they're going to have that ose ending. So examples, triose has 3 carbons, pentose 5 carbons, hexose 6 carbons. And I'm not expecting you to memorize all these things about it. You just need to be able to look at a picture and identify that that is a monosaccharide, that is a carbohydrate. Okay, so another feature about our monosaccharides is they can be either linear, like on our previous slide, or they can be ring-shaped. And they're going to be ring-shaped most of the time in aqueous solutions, which will most of the time be our cells, because we're made of mostly water. Okay, so when it's in a ring structure, see here how we have this linear, it's wrapping around and creating this ring. This is what our ring is going to look like. It's still carbon, hydrogen, oxygen in a ring, but it is not linear anymore. And they can be 5-membered rings or they can be 6-membered rings. But these are all examples of monomers of our monosaccharides, of our carbohydrates. Okay, here's some other examples. Maltose, lactose, sucrose. Again, some examples. We can refer to these as disaccharides because they are made of 2. So just another term relating to carbohydrates. And then polysaccharides is when we have long chains of monosaccharides, and they can be branched or unbranched, and they can have different types of monosaccharides in them. They can have 5-membered rings and 6-membered rings. It doesn't matter. They're all going to be monosaccharides. So here's an example of glycogen, which are these black granules, and that is a form of energy that is being stored for these flatworms. So we can have linear structures, we can have different spirals and branching structures, and they're all made of these monomers. So these are all examples of how we can have different structures of our polysaccharides. Okay, so here again, showing this long linear polysaccharide is just a bunch of these monosaccharides bonded together. Okay, next is our lipids. Reminder, our lipid monomer is our glycerol and fatty acid. Polymer is lipids. Function is long-term energy storage. It's also used in membranes. Its structure, carbon, hydrogen, oxygen, and sometimes phosphorus, which we will talk about right now. So here's the overall structure where we have glycerol and then our fatty acids. Okay, big thing about our lipids is that they are non-polar, which means that they have different poles, just like our water molecule, and so therefore they are going, our water molecule has the poles, so these are opposite as ours, so they will be water-fearing, they will be hydrophobic. So an example in real life is that there are hydrophobic lipids on the fur of aquatic mammals to protect them, which is kind of cool. Okay, we have saturated and unsaturated fatty acids. So our saturated acids here and our unsaturated. Saturated are going to have single bonds. They will be solid at room temperature. Butter is an example of a saturated fatty acid. Saturated, they're saturated with hydrogens. They have as many hydrogens as they possibly can have. Our unsaturated fatty acids here, they're going to have a double bond somewhere. Sometimes they have more than one, but at least one double bond. That makes a kink in their tail, and that kink makes it difficult for those molecules to pack together, which means that it's more difficult for them to be solid, so they're going to actually be liquid at room temperature. So this is going to be our oils. If you use like can't believe it's not butter, like those butter alternatives, those are going to be our unsaturated fats, which will be liquid at room temperature. Okay, our phospholipids, this is where we have our phosphorus involved. Okay, so we still have our lipid, we have our tails here, glycerol, and then the phosphate group is attached. And so notice our phosphate group has a charge here, and that charge is going to make it polar, right? So we now have non-polar tails and then a polar head. So it now has non-polar and polar regions. So we have a hydrophobic tail and a hydrophilic head. Okay, so hydrophobic and hydrophilic. So think about our cells are made of water. So we have one of these guys just floating around. There's water, and it doesn't like water. This hydrophobic part doesn't like water. The hydrophilic part doesn't mind, but hydrophobic wants to get away from that water. So it will move away from that water. But our cells don't just have one water molecule. They have water all over the place, and there's lots of these phospholipids. So think about and try to draw how these phospholipids would arrange themselves to minimize the amount of phospholipid that is this hydrophobic part touching the water. Okay, so what you should kind of come up with is something like this. Maybe these circles where everything on the outside is the hydrophilic head, and then the hydrophobic tails are inside, away from the water around them. Or we get these layers like this where there's water on either side, but that's where the hydrophilic head is, and the hydrophobic tails are protected inside. This is where we have our membranes, and this is exactly what our membrane looks like. Hydrophobic region and our hydrophilic regions. Okay, so now let's go to our proteins. Proteins, our monomer is amino acids, our polymer, polypeptides, our bond name is a peptide bond, and the function, it has all sorts of functions, structure, enzyme, all sorts of things. And its structure, this is a structure we haven't talked about a whole lot, but we talked about it having carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. But it will have, this is the main amino acid structure. We have a nitrogen, a carbon, and then another carbon. This nitrogen is called an amino group, this NH2. This carbon is double bonded to one oxygen and has an OH group on this side. It's called a carboxylic acid or a carbonyl group. And then this carbon in the middle is called our alpha carbon, and off of it we have a side chain. R is not an element on the periodic table. This is just meaning it can be any sort of different thing attached onto that spot. Okay, so here's some examples. See, we have our NCC, NCC. So these are all amino acids, but that R is changing to be different things. And depending on the type of R group, it depends on the function, how it's related to the structure of our amino acids. So we can have some that are hydrophobic, and we have ones that are hydrophilic or ionic. So notice here, this is a hydrophobic. It only has carbon and hydrogen. It's going to equally share those electrons. But remember, oxygen is really electronegative. It's really greedy. It wants those electrons. So they're going to pull closer to oxygen, making it have a little bit of a charge, making this have a charge, which will make it hydrophilic. And then we can also have ionic. So the big thing you're looking for is if it's just carbons or if it has oxygen or other elements involved. Then it's going to be hydrophilic or ionic. And ionic is going to interact similarly to hydrophilic because of the charge. Okay, so think about where in the cell then, with water all around it, where would those hydrophobic amino acids be located? And where would the hydrophilic amino acids be located? And you can think about the waters all around it. We're going to want to keep those hydrophobic ones away from the water. So it could be inside of the protein so that it's protected, or it could be inside of our membrane, right? There's a hydrophobic region right here. So we can have proteins that go through membranes where here we have our hydrophobic amino acids in the chain, and then our hydrophilic amino acids are out here in the water on either side of the membrane. Okay, our last one is our nucleic acids. Our monomer are nucleotides, polymer, RNA and DNA, bond name is our phosphodiester bond. Its function is to store genetic information, and its structure, which we haven't talked in super detail about, is a 5-carbon sugar, a phosphate group, and a nitrogen base. Has this overall structure. Phosphate group, 5-carbon sugar. So it actually is made of part of our carbohydrates, right? It's got a sugar in it, but it also has this phosphate group and a nitrogen base. Okay, DNA versus RNA. There's a couple differences. Remember that you might remember that DNA stands for deoxyribonucleic acid and RNA is ribonucleic acid. So the sugar is actually different in either of them. One uses ribose and the other uses deoxyribose, and they're both 5-carbon sugars. And so one is our ribose and one is our deoxyribose. So look at them. What is different between these two molecules? They're almost exactly the same. And what is different is that our deoxyribose has lost one of the oxygens. It's lost this oxygen down here at the bottom. Everything else is exactly the same. And that one little oxygen makes a huge difference on whether it is DNA or RNA. It makes a huge difference in its function. Okay, so here again is the basic structure of our nucleic acids. So they would all have the phosphate group, but the 5-carbon sugar is going to be different, deoxyribose and ribose. So which one's RNA and which one's DNA? Our DNA is the deoxyribose and our RNA is the ribose. Okay, another difference between our DNA and RNA is that nitrogen base, this guy right here. So a lot of them can be the same, but there's some that are slightly different. So we have adenine, guanine, cytosine, and thymine are the options for our nitrogen bases here, and you probably remember this from biology or from the past, A, G, C, and T. Our RNA, on the other hand, have different options. It can have adenine, guanine, cytosine, so these top 3 are the same, but instead of thymine, it's going to have uracil, and we'll eventually talk into how the structure of these are different. Okay, another difference between the two is the strandedness. So our DNA is double-stranded, and our RNA is mostly going to be single-stranded. It can also have double-stranded areas. It can also have areas where it's double-stranded and then goes to single-stranded and then back to double-stranded and then back to single-stranded. So it can have really more complicated structures. Okay, so here is an example of a question you might see on the AP test talking about our different proteins. So this is talking about different molecules, and so you can notice what we have here about which ones are hydrophobic and which ones are hydrophilic. So pause this and see if you can figure them out. Okay, so the answer is D here. So here molecule C is showing a lipid, a phospholipid, where we have that hydrophilic head and those hydrophobic tails. So that means we have a hydrophobic region right here. So C is made up of hydrophobic and hydrophilic regions. B is going through that membrane. So in the middle, we're going to have hydrophobic amino acids, but on the ends, we'll have hydrophilic amino acids. And then this molecule is all out in the open. It's all in the water. So it's going to be pretty much all hydrophilic. So D here explains this the best. Okay, so the rest of class, we worked on the sorting. We only got through today through the protein part. So try your best on that, and we'll work on this more next time. Bye, guys.